So Professor Babiloni is a cherished friend of Mind Lifeline. And I actually still remember your talk from Mindscapes conference from last June, and I'm sure that everyone here will remember today's talk. He is leading the Laboratory of Neuroscience of Human Higher Functions at Sapienza University of Rome. His expertise lies in brain rhythm research associated with neurodegenerative diseases and age-related cognitive decline. He employs neuroimaging and EEG to examine irregularities in conditions such as Parkinson's and Alzheimer's, notably contributing to the PDWAVES Consortium and the Alzheimer's Association International Society Advisory Board. His presentation promises very important insights into a particular area of Alzheimer's. We are deeply grateful for your participation. Professor Babiloni, you are invited to begin.
Thanks for this nice introduction and for your nice words. And okay. Okay, you can see in this slide my co-workers who contributed to this presentation and results. And thanks a lot for the organizers for inviting me in this conference. Of course, today the previous speakers talk about dementia, and dementia is defined as severe cognitive deficits and disabilities in the activities of daily living, which is wearing herself or shopping or preparing meals and so on. And you can have different causes to induce dementia and cognitive deficits in aging. And you see in this graph that there are some neurodegenerative diseases which are progressive and induce continuous decline of cognitive functions and abilities in daily living. And you see that Alzheimer's disease and dementia with Lewy bodies, frontotemporal dementia, and Parkinson disease are part of these neurodegenerative diseases. And today we will focus on Alzheimer's disease. And Alzheimer's disease is characterized by two pathological, abnormal accumulation of proteins, of two specific proteins in the brain. And you see in this slide, they are colored in blue and orange. And in blue, you have amyloid beta proteins. And you see that they are accumulated in the brain in the intermediate stages of Alzheimer's disease. You see in the frontal lobe and in the medial parietal cortex, and in the stages of dementia and these severe cognitive deficits and disabilities in the activities of daily living, you see this increase in accumulation in blue, but also overlapping with orange color that characterizes the accumulation of another protein, tau, phosphorylate tau in the brain. And you see from preclinical stages and the intermediate stages of my cognitive impairment, and then severe stages of dementia, that you have a spread of these abnormal accumulation of these two proteins. These are the neurobiological causes of Alzheimer's disease. And you see that the main focus in the clinical evaluation of the patients with Alzheimer's disease is the measure measurement of cognitive functions. And you see in the left some domains of these cognitive functions. And you see that you can measure in Alzheimer's disease decline in thinking abilities that interfere with everyday life. And you see also significant memory loss in Alzheimer's disease in most cases. And so you see that also other cognitive deficits are possible or probable. Planning, activities in the daily living, or spatial orientation, and so on. And you see that in the clinical evaluation of the manifestations of Alzheimer's disease, you cannot find the evaluation of vigilance regulation. So the ability of the brain to maintain a certain degree of vigilance. And this is a really important higher function in the brain, because if you are not able to maintain a good, appropriate stage of vigilance, you cannot follow social conversation, you cannot follow TV programs, and so on. And because you risk to go to drowsiness and lowering of vigilance. But and so it is frequent that you see patients with mild dementia due to Alzheimer's disease have some drowsiness during daytime and go to sleep in the afternoon, for example, or in the early morning. But unfortunately, the clinical evaluation of Alzheimer's disease do not probe, do not measure vigilance. And the regulation of vigilance is something very important for consciousness. In this graph, you see how we can investigate the consciousness. You see that consciousness can be evaluated in vertical axis in the sense of level of vigilance, level of consciousness, from full arousal, full vigilance, to intermediate stage of vigilance, or drowsiness or sleep in the vertical axis. And in the horizontal axis, you have the vividness of the contents of consciousness, the representation, mental imagery, thinking, and all kind of mental objects we have in our flow of consciousness. And you see that in this graph, you can put different stages of consciousness and vigilance, from alertness to drowsiness, light sleep, deep sleep, and this transition is important also in patients with Alzheimer's disease, and is not measured with biomarkers in the actual guidelines for the evaluation of Alzheimer's disease. In contrast, the regulation of vigilance and the consciousness level is very important. And Alexander Luria, a genius of neurosciences in the previous century, developed a theory of the working brain, and he conceptualized 3 different units of the brain underpinning higher functions. And the first unit was involved in the sleep wakefulness cycle and in modulating the vigilance consciousness level. And you say that in gray color, you have the brainstem and diencephalon. And then these black arrows are the connections between the thalamus and the basal forebrain and the cerebral cortex. Continuously, these ascending, arousing systems maintain the appropriate cortical arousal, underpinning the maintenance of vigilance, allowing you to be lucid in the mind and to follow what is happening. The second unit is represented in the posterior cerebral cortex and in limbic system. You see here hippocampus, amygdala, and you have also other components of limbic system in anterior cingulate, for example. And these limbic systems are important for emotional coloring of the external stimuli or internal stimuli, endogenous stimuli. And so perception, episodic memory, and emotions are regulated by this second unit of the brain. And the third unit is represented in the prefrontal cortex in gray color here, and underpins motivation, executive frontal function, attention, and the internal language, which regulate our thinking, our planning, and so on. So the focus of the research of my group and other groups are to exploit the first unit of the brain in the evaluation of brain functions in Alzheimer's disease patients. The basic methodological approach to investigate the neural basis of a vigilance and the consciousness level in Alzheimer's disease can be based on the measures of brain activity, for example, like electroencephalographic activity on functional magnetic resonance imaging. And you can also investigate the structure of the brain underpinning vigilance consciousness level by means of structural magnetic resonance imaging and the connections in the brain from a volumetric point of view. And then another approach is the causality by means of brain stimulation by drugs or non-pharmacological intervention, or brain stimulation with transcranial magnetic stimulation, for example. In our study, we evaluate electroencephalographic activity as functional measures of brain activity. And you see that it's very easy to investigate the regulation of vigilance levels, of consciousness levels, by means of electrode cap over the scalp and collecting electrical brain activity and visualize the electrical brain activity in the form of electroencephalographic waves in the screen. And the subjects, we ask the subject to stay in a resting state condition, quiet wakefulness. And the instruction are that he had to stay 3 or 4 minutes with eyes closed, quiet vigilance and wakefulness. And then we can also ask to open the eyes and to stay in the resting state, eyes open. And so in this kind of experiments, we can see the transitions of vigilance from consciousness to drowsiness and light sleep, and in some cases, even deep sleep. When you see this symbol, you see persons with eyes closed, resting state. And you see that you can, with the scalp electrodes, you can record EEG activity in the form of waveforms of electrical activity in the scalp. And when the subject is in the resting state condition, you can record in the wakefulness, quiet wakefulness, a dominant oscillation of electroencephalographic activity at about 8-12 hertz, 8-12 cycles in a second. And when there is some transition from quiet vigilance to drowsiness, you see that you have fluttering of EEG activity. And in the very early stage of sleep, stage 1 of sleep, you have this full fluttering, or you can see oscillations at a slower frequencies in the theta range, less than 7 hertz. And you see that you have the transition to deeper stages of sleep with characteristic waveforms. So this is to stress that EEG activity is perfect to capture the transitions of vigilance from quiet wakefulness and vigilance to drowsiness and light sleep. And when you see here Vincenzo Crunelli in Cardiff, in Scotland, developed some fantastic research activities in cats and mice to investigate the generation of electroencephalographic activity based on spiking activities in neurons in the thalamus and in the cerebral cortex in tissues. And he could demonstrate that, for example, in thalamic recordings of local field potentials, you can see alpha rings around 8-10 hertz. And these oscillations are related to spiking activity, which is represented by dots, blue dots, in the upper part of the graph. And you see that when you have a more dense frequency of spiking activity in thalamic neurons, you can record in the local field potentials alpha rings. And the same is true in the from neurons of the cerebral cortex. And so, in other words, Vincenzo Crunelli could demonstrate that alpha rings in the cerebral cortex and the thalamus are related to circuits between thalamocortical neurons and corticothalamic neurons. And you see this in the graph. And that an ample alpha ring is important to regulate cortical arousal. Okay, how can we measure these electroencephalographic activity and alpha activity to produce biomarkers? And a very simple way is the spectral analysis of EEG rhythms. In this graph, you see here in delta plus alpha plus beta, a sketch, an example of a schematic EEG activity. It's formed by 3 oscillatory electrical activities, one in the delta range, 1.5 hertz, another in the alpha range, 8 hertz, and another in the beta range, 20 hertz. And you see that when the spectral analysis is able to summate this elementary oscillatory activity to generate a signal, and can also perform the reverse solution, can start from recorded real true signal, electroencephalographic signal, and can decompose in elementary oscillations. And another important measure of this approach is the creation of EG power spectrum in the down part of the graph. And so in the EG power spectrum, you can observe the representation of the amplitude of oscillations, of the elementary oscillations, squared, so the power. And so, for example, you see that alpha amplitude is higher than delta amplitude and beta amplitude in this example. And you see that in the EG power spectrum, alpha power is higher than beta power and is higher than delta power. And this is exactly the kind of representation of our studies. Our methodological approach, is that we start with the recording of resting state, for example, eyes closed, and you see the EG activity in the scalp electrodes, and we select the periods artifact-free without high movement artifacts or muscle artifacts or head movement artifacts. We select the periods artifact-free and use these periods as an input for the estimation of the cortical sources of scalp EG activity. And Iloreta freeware is available in the public domain, and you can use Iloreta to estimate the cortical sources of EG activity. Due to the low spatial resolution of our electrode montage, we use large cortical regions of interest in red in the cortical model of Iloreta sources, and we average the solutions within these large regions of interest: the frontal, central, parietal, temporal, occipital, and limbic. And so you will see our graphs of cortical source activity in these large cortical regions of interest. And this is exactly what you see in this graph. In this experiment, we wanted to demonstrate that the cortical source activity from a resting state EEG is related to vigilance level. And so we perform this experiment in the healthy subjects in two recordings. The first recording is a normal resting state EEG recording in blue. And you see that the maximum source activity is in parietal occipital and is in the alpha frequency, the dominant posterior alpha rins in the resting state, eyes closed, as a measure of quiet vigilance regulation of the cortical arousal. But the same subjects were recorded after 1 night of sleep deprivation in red. 1 night of sleep deprivation means that you do not sleep during the night. So when you receive this recording, you are a bit tired, with some fatigue in the mind, a low level of vigilance. And you see that even if you are awake in the resting state, eyes closed in red, after 1 night of sleep deprivation, the alpha source activity is reduced in several cortical regions. So this is a demonstration that alpha rins are related to regulation of vigilance level, what we want to do in Alzheimer's disease. So in another experiment, we focus on patients with Alzheimer's disease in red with mild cognitive impairment, so the intermediate level of the disease. And we could have the diagnosis by means of cerebrospinal fluid biomarkers of Alzheimer's disease. You see these lumbar puncture in the spinal cord, and you can measure a beta amyloid and phospho-tau in the cerebrospinal fluid. And if you see some levels, you can say, okay, this person is an Alzheimer's disease patient. And then with neuropsychological test, you can see that there is a mild cognitive impairment, not dementia, not yet a dementia, because the patient is able to prepare meals, is able to wear herself, is able to do shopping, and so on. And we compared the EEG source activity in Alzheimer's disease mild cognitive impairment with a control group of mild cognitive impairment not due to Alzheimer's disease in blue. And you see in the graph in blue the non-Alzheimer's disease mild cognitive impairment, in the red Alzheimer's disease with mild cognitive impairment. And you can clearly see that Alzheimer's disease mild cognitive impairment show at the group level lower alpha source activity and higher delta source activity in the temporal source region. So this is a nice demonstration that Alzheimer's disease patients have an additional vigilance dysfunction in the stage of mild cognitive impairment when compared with other patients with mild cognitive impairment, same cognitive deficits, but due to different diseases. Another demonstration that Alzheimer's disease patients have characteristic vigilance dysregulations as revealed by EEG data was shown by this experiment. In this experiment, we compared the resting state EEG source activity in closed in a group of Alzheimer's disease mild cognitive impairment in red, a group of Parkinson disease, another disease with mild cognitive impairment in green, and control healthy normal subjects and old in blue. And again, you see that the higher source activity in the alpha range is in blue, is in the healthy subjects, normal subjects. And we have a very significant decrease in cortical source activity in parietal, occipital, temporal, and limbic in Alzheimer's disease mild cognitive impairment in the red. In Parkinson's disease with mild cognitive impairment, we have a decrease of alpha rims in the sources, but not so great as Alzheimer's disease MCI. And you can see that you have also an increase in delta source activity in Alzheimer's and Parkinson's in temporal sources, but without differences. But in parietal, the abnormal increase in delta source activity is higher in Parkinson's disease with MCI in relation to Alzheimer's disease with MCI. So you see that these vigilance, these regulations are different in patients with different disease, Alzheimer's and Parkinson, even with the same cognitive status of mild cognitive impairment. So vigilance is something different from cognition in some sense. And it's not sufficient to use neuropsychological tests to measure cognitive deficits, because you need also EEG to measure vigilance dysregulations. And another demonstration of the importance of vigilance transitions and dysregulation in Alzheimer's disease my cognitive impairment is shown in this experiment. In this experiment, we distinguish the transitions from wakefulness to light sleep, stage 1 of sleep. And we classify the wakefulness with alpha dominant in the posterior regions, and the light sleep with fluttering and the ripples. And you see that we divided in the stages of eyes open, resting state, full vigilance, wakefulness in the eyes closed, so wakefulness but low vigilance, fluttering, very light sleep, and ripples, light sleep. And if you see frontal, parietal, and occipital regions of interest, you see the frequency delta, theta, alpha 1, alpha 2, alpha 3. And you see that in the resting state, eyes closed, wakefulness, you have in red, more delta in the frontal areas. You have more delta also in the fluttering light sleep, and also more delta in Alzheimer's in ripples light sleep in relation to blue normal subjects. And when you see in the occipital areas, you see more alpha in blue in healthy subjects, less alpha in Alzheimer MCI, in resting state, eyes closed, wakefulness. And then you have no difference in fluttering and very few difference in ripples in some cases. So you see that it's nice that in the transitions you have some abnormal delta in Alzheimer's disease and low alpha in resting state, eyes closed, wakefulness. The vigilance is abnormal in Alzheimer's disease MCI patients, also in relation to the years of education. And you see, for example, in this experiment, we have a healthy, normal old subjects in the upper part in blue, normal with less education, light blue, and the normal with plus education in deep blue. And you see that you have a different in alpha source activity, resting state, eyes closed. More alpha in more educated, healthy subjects. So instructions empower the maintenance of vigilance, and this is reasonable. But in the patients in Alzheimer's disease, my cognitive impairment, you see that you have differences in the sense that patients with more education have less alpha. And this means that this more education compensate the disease progression and the impact on vigilance level. So more educated Alzheimer MCI are able to maintain the cognitive function in relation to Alzheimer MCI patients with less education. And so a compensative effect of education on vigilance. And we see the same compensatory role of education in the preclinical stage of Alzheimer's disease, the stage that we call subjective memory complaints. The subject think I have some memory problem, but in the neuropsychological test of memory, the score is normal. And in these patients, we have Alzheimer's disease pathology in the brain as revealed with amyloid PET in this experiment. And we could show that there is this compensatory activity because in the subjective memory complaint positive to Alzheimer's disease, you can see less alpha source activity in comparison with subjective memory complaint positive to Alzheimer's disease, but with less education attainment in blue. So compensatory activity and role of education. And in healthy participants with subjective memory complaint, negative to Alzheimer's disease biomarkers with a PET, you see that more educated people have this neuroprotective role of education with more alpha in red compared to the control group with less education in blue. So similar to Alzheimer's disease MCI experiment. Okay, one issue is if this biomarker, e.g. biomarker of vigilance, can also be sensitive to the progression of the disease. And in this experiment, we tested a group of Alzheimer's disease cognitive impairment patients during 1 year. And so you see the first recording in blue and the second recording in red. And you see that in the first recording, you have some levels of source activity in alpha range in the parietal, occipital, and temporal cortical regions of interest, the resting state, eyes closed. And after 1 year, in the group show a very clear decline of alpha source activity in red. And so this means that EEG biomarkers are sensitive to the progression of Alzheimer's disease in 1 year in patients with mild cognitive impairment. And so you can use these to see the impact of the disease on the vigilance regulation. In another experiment, we repeated the same experiment in patients with Alzheimer's disease dementia, so the dementia stage of the disease. And again, you see the first recording in blue and the second recording after 1 year in red. And you see that you have a significant decrease of alpha in red and increase in delta from resting state EEG after 1 year, even in patients with dementia. So you can continue to use EEG recordings even in the dementia stages. And can you tell me if I have additional minutes or not?
Yes, sure.
Yeah, and how many minutes I have?
So we have like 3 questions until now from participants. So we just have to answer those questions and like the rest of your talk. Maybe 5 to
5
7
minutes. Okay, good, good.
Okay?
5 minutes are enough. Thank you,
Okay.
Lea. So another approach is to put in relation neuroimaging with EEG biomarkers. And for example, the first structure of interest is the cholinergic system in the brain, because you know that Alzheimer's disease have some deterioration of the cholinergic system in the brain, and we give cholinergic therapy with acetylcholinesterase drugs to improve cholinergic system. And you can measure the lesions, the impairment in the cholinergic systems in the magnetic resonance imaging in these masks of red, orange, and green, which are the parts of the brain in which you can find the tracts from cholinergic basal forebrain to cerebral cortex. And we did this measure in magnetic resonance imaging in this experiment. And we could stratify Alzheimer's disease MCI patients in cholinergic plus impairment and cholinergic less impairment. And you see the results in this figure. And you see that in the healthy subjects, you have in red high sources of alpha RIMS. a decline in Alzheimer's disease patients with less cholinergic impairment, but in Alzheimer's MCI with plus cholinergic impairment, the reduction of alpha source activities is greater, more abnormality, more vigilance dysregulation in relation to cholinergic lesions. And another demonstration of the importance of cholinergic system is in this experiment in which we investigated the EEG activity before and after 1 year of cholinergic therapy in Alzheimer's disease patients. Resting state, eyes closed, EEG source estimation in the regions of interest, in the frequency bands, and the subtraction of the results of the second recording after 1 year of treatment and baseline first recording. So it means that the zero difference means the same source activity, no changes after 1 year. But if you see negative values in the down part of the graph, you see that you have lower source activity in some frequencies. And this is the case of alpha. And you see that you have lower source activity after 1 year of treatment in the two subgroups of Alzheimer's disease dementia patients. In red, the patients not responding to the therapy. This means that they have some cognitive deficits increase after 1 year. In blue, patients that were stable with cognition after 1 year, so good response to cholinergic therapy. But you see that even patients responding to the therapy, having stable cognition, have a decrease of alpha source activity in blue. And this means that the disease is running in any case. But this is a demonstration that the cholinergic system react to the therapy. So you have less reduction of alpha source activity in patients responding to the cholinergic therapy. And in this study, we put in relation the atrophy in the cerebral cortex, normalize gray matter volume in the cerebral cortex in the horizontal axis in relation to alpha source activity or delta source activity in the resting state, eyes closed. And you see that any participant is a circle. And you have here subjects with normal, healthy cognition, mild cognitive impairment, and dementia due to Alzheimer's disease. And you see that there is some correlation. The higher the pathological delta source activity, the lower is the brain cerebral cortex volume. The higher the alpha source activity, the higher the cortical gray matter volume. So a strict relation between a neurodegeneration cortical atrophy and EEG sources. And other demonstrations of this structural relation is in this publication, in which we could demonstrate that the atrophy in default mode network, you say that in gray, we have Alzheimer's disease patients. In black, healthy subjects, you see the decrease of the gray matter volume in the default mode network is related to decrease of alpha source activity. And is a correlation with the regressive modeling. And you see that you have atrophy also in sensory motor network in the cerebral cortex and in dorsal attention system. But the correlation in posterior inferior cerebral cortex, the decrease of alpha, circle Alzheimer's disease dementia, triangle healthy subject and all, you see that this decrease is correlated with the atrophy in the fold mode network. Okay, I see that I finish my time. We have also some association with functional magnetic resonance imaging connectivity, but we come to the conclusion that alpha source activity underpin vigilance consciousness level, and we could demonstrate this in relation to sleep deprivation, cholinergic deficit, fold mode network. So we propose that EEG biomarkers are the pathophysiology biomarkers in the framework table of amyloid biomarker with CSF and PET, tauopathy, CSF and PET, and then pathophysiology with EEG to measure vigilance deregulations and dysfunctions, and then neurodegenerative biomarkers with neuroimaging. Thank you for your attention, and sorry for the lengthiness of my presentation. These are my co-workers.
Thank you very much, Professor, and don't worry at all. We enjoyed it very much, and actually there are a lot of questions. So I will start with the first one from Catalina. Thank you, Professor Babiloni, for your presentation. My question is, how might the findings on neurosynchronization at delta and alpha frequencies contribute to our understanding of sleep disturbances and circadian rhythm disruptions, often observed in individuals with cognitive impairments?
Yes, thanks for this question. You are right that I emphasize the alpha source activity in the resting state as close, and I didn't discuss a lot the increase in delta and theta. And you are right that delta is pathological in the resting state EEG and the transition to light sleep. But in our results, I didn't present the data with metabolic PET, in which we could demonstrate a relation between metabolic PET decrease and increase of EEG delta source activity, and other data with increase of delta in relation to neurodegeneration. So delta is important with abnormalities also in vigilance, but is more sensitive to cortical neurodegeneration and the loss of metabolic activity. And so in this sense, alpha is most sensitive to the impact on the vigilance neural systems. And this is why I emphasize alpha, but I agree that also delta and theta can get into artificial intelligence, machine learning, to have some profile of a neural system of vigilance. So I agree with you.
Okay, thank you. The next question is from Laura. She's saying, a very interesting presentation, Professor. Thank you for all the information. I do have one curiosity. Given the decreased vigilancy that people have due to sleep deprivation, can we say that prolonged sleep deprivation gives a similar effect on vigilance as Alzheimer does?
Yeah, you are very sharp to see that we could reproduce abnormalities in the vigilance regulation in healthy adults with 1 night of sleep deprivation. So one can say, but we have actually the reproduction of a sort of Alzheimer's disease in healthy subjects. In reality, in the graph, you could see that alpha was decreased in all cortical source regions everywhere after 1 night of sleep deprivation. In contrast, in Alzheimer's disease, you have this kind of dysfunctions mainly in the posterior cortical regions. So the topography is a bit different between the effect of sleep deprivation in healthy subjects and Alzheimer's disease neuropathology. But you are right that you can use EEG recordings and analysis also for other diseases or other conditions if you need a biomarker of the neurophysiological oscillatory mechanism underpinning vigilance regulation. You're right.
Okay, thank you. The next question is from Marij. I hope I pronounced the name right. So, hi, Professor Babiloni. Thanks for the enlightening lecture. I am wondering if similar compensatory effects of education could be observed in people suffering from post-COVID cognitive problems.
Exactly. We now are publishing some results of resting state source activity in long COVID-19 patients. And we could observe that they have less alpha source activity in relation to mental fatigue. And one point is that when we measure education attainment, we see that COVID-19 post-COVID patients had less education, less protection. And so it's incredible that educational attainment can protect you by long COVID-19, probably, the long COVID-19. So it's an important variable to investigate in the future.
Okay, thank you. Another question also from Marij. What happens to the EEG correlation graphs for healthy and diseased subjects when filtering the outliers?
Okay, you are right that outliers are very important, and our group manages outliers using log10 transformation of EEG source activities. So when you use log10, you reduce the skewness of the data, and really you do not see a significant number of outliers. So my suggestion to use this kind of normalization and data compression to avoid outliers. And but if you have outliers, even with log10 transformation, you can use the GRABS test, the statistical test, to define a statistical threshold and accept only individual values under the threshold, the statistical threshold you impose. So please treat statistically the issue of outliers in your experiments.
Okay. So one more question from Alexandru. Hello, Professor. Thank you for this very insightful presentation. I wanted to ask, in your opinion, in what situations do these methods lack in offering enough information about Alzheimer's, and what would be the solution to fix this aspect?
Yeah, the point is what I called in two editorials the dark side of the precision medicine in Alzheimer's disease. The point is that if you want to obtain the diagnosis of Alzheimer's disease, you don't need EEG or magnetoencephalography or transcranial magnetic stimulation. You don't need this because you have to measure amyloidosis and tauopathy in the brain. And EEG is not a measure of amyloidosis or tauopathy. But in the precision medicine, you want to see if there is something wrong in the memory system, regardless the relation to amyloidosis and tauopathy. You want to measure memory or executive functions or visual spatial activity, because you want to see what is the progression of the disease and if you can stop the disease. And so you use neuropsychological tests, even if they do not measure amyloidosis and tauopathy. Even if you use this test with Parkinson dementia or with Lewy body dementia. And the same is true for EEG. You have to use EEG even if you cannot obtain the diagnosis of Alzheimer's disease, but you can obtain an objective measure of vigilance dysfunction. And you can use this number, this biomarker, to see if your treatment is able to improve the vigilance regulation. If the subject do not fall asleep during the time in front of TV programs, or while he is reading the newspaper or have a social conversation. So the point is that in the precision medicine, you want to have biomarkers for the symptoms, for the dysfunctions, in order to see if your treatment is able to normalize the dysfunctions. And EEG is perfect for measuring the neurophysiological underpinning of vigilance as an endpoint of the therapy.
Okay, thank you. We have an additional question from Catalina. Could the distinctive patterns of cortical neuron synchronization observed in this study potentially serve as biomarkers for distinguishing between different cognitive disorders, including Alzheimer's, in clinical settings?
Yeah, my opinion is that the diagnosis should be neurobiological. So if you have a Parkinson, you have to test alpha-synucleinopathy. We don't have this test now, but we can measure the uptake of dopamine in the dopaminergic systems with the PET or SPECT. You can measure amyloidosis and tauopathy in the cerebrospinal fluid or with the PET. And we have to go to neurobiology biomarkers for the etiological diagnosis of these neurodegenerative diseases. But we need EEG as a biomarker of a neurophysiological underpinnings of vigilance and to improve vigilance in our patients. Parkinson, Lewy body, frontotemporal, Alzheimer's, regardless the diagnosis, is the evaluation of dysfunctions, the same use of a neuropsychological test for memory, attention, and language. You can use this even if you have memory deficits in Parkinson disease with dementia, Alzheimer's disease with dementia, or Lewy body dementia, or mild cognitive impairment, amnesic due to Parkinson or Alzheimer's. You use this test even if you cannot have the neurobiological diagnosis, and you can use EEG even if it's not a direct measure of alpha-synucleinopathy or amyloid or tau or TPT43 and so on.
Okay.
This is my message. This is my message.
Sorry, I interrupted. So the last question is from Sabina. Hello, thank you, Professor Babiloni, for this interesting talk. What do you think about using AI to interpret EEG or fMRI exploration, result interpretation, and data correlation?
Yeah, I'm working in this field, which is the future, of course, because we ideally could use a lot of biomarkers together to have a composite frailty score of the brain status. And we can use these to see what is happening in the patient over time. So we can reconstruct the relation with artificial intelligence, machine learning tools. But my suggestion then to follow the new wave of this field with explainable artificial intelligence tools that are able to inform you if you use 10, 20 variables, which are the variables more important to predict the cognitive decline or to produce a cognitive status score or brain status score. And the explainable artificial intelligence tool can say, okay, you use 20 variables, but the very important ones are variable 2, 5, 8, 10. And when you see and read these variables, you can say, okay, this is a good computation because these variables are sensitive to the disease. This is my point.
Thank you very much. Thank you for answering all the questions, Professor. And we really appreciate everything that you have offered today.
Thank you very much. Thank you very much for your invitation. I love this initiative, and I hope that we can meet again soon. Bye.
Thank you.
Bye.